Guys, welcome you all. On behalf of DBMCI, I would like to welcome you all and wish you a very good luck. In this session, I'll be talking about a little bit about one of the amino acid. The purpose of this session is to give you more insight of how basics are important. My mentor always used to tell me, see Varsha, if you know the reason, ठीक है, कि कोई भी चीज़ क्यों हुई है उसका कारण अगर पता होगा तो उसके कॉन्सिक्वेंसेस भी पता होंगे. फॉर एवरी डिजीज़ देयर इज़ एन अंडरलाइंग मैकेनिज्म. ठीक है, अगर वो अंडरलाइंग मैकेनिज्म इज़ व्हाट बायोकेमिस्ट्री इज़, और वही हम सेशन ले रहे हैं बायोकेमिस्ट्री का ही, माई सेल्फ डॉक्टर वर्षा. अगर हमें पता होगा कि कोई भी डिजीज़ क्यों हो रही है तो उसके कॉन्सिक्वेंसेस, उसके सिम्टम्स को हम उससे कोरिलेट करके वी कैन गेट एन बेटर आइडिया. कितना क्रैमिंग करेंगे, देयर आर 19 सब्जेक्ट्स. ठीक है, कितना क्रैमिंग करेंगे, जितना समझ कर पाएँ, एक सीरीज़ हमें पता चल जाएगी तो हम कोरिलेट कर सकते हैं. ये अच्छा ऐसा है तो इसका मतलब ये होने वाला है और ये ऐसा होगा, राइट. So we can pick the best answer in the question. Okay, please feel free to drop me an email if you feel there is something I can give you. If you want me to release some video on a topic you want, you are most welcome. So I'm talking about tryptophan here. Tryptophan is one of the aromatic amino acids. See, we have 3 aromatic amino acids: phenylalanine, tyrosine and tryptophan. Okay, tyrosine and phenylalanine, the only difference is the hydroxyl group down there. Okay, so phenylalanine, from phenylalanine we can synthesize the tyrosine, right, or tyrosine can be synthesized from the phenylalanine, and tryptophan is another one. So they have the aromatic rings. This kind of a ring that is there in the phenylalanine is kind of a phenyl ring or phenol ring. Okay, same thing, tyrosine is here. Can we say it phenol ring? No, we cannot say it phenol ring, though it might not be asked, but we should be knowing. So if it has OH only, then this is a phenol ring, right? And tryptophan, what we have a 2 kind of ring system. One is 6-membered system and another is a 5-membered ring. Okay, and that 5-membered ring, has a nitrogen here and a double bond. This is the circular one and this is a double bond. Okay, this what we call is an indole ring. So it has a benzyl part. And what is this 5-membered? For 5-membered is for furan ring, right? It gives you what? The indole ring altogether. So this is what we will be focusing on. Now, these ring kind of structures are very difficult to make by the body. So all these come under the essential amino acid. Essential. All aromatic are the essential amino acid except tyrosine. Right? Why I said this way? Why I said it other way around? Because tyrosine can be synthesized from the phenylalanine. So this is not considered as an essential amino acid. This is essential amino acid only when the phenylalanine is lagging. Okay, it is not there. Then you have to supplement the tyrosine in the food supplement or by the food materials. There should be more intake of the tyrosine now. So out of 3 aromatic amino acids, we know these 2, the phenylalanine and tryptophan is the essential amino acid. What are essential amino acids? Those which should be in the diet, right? Because they cannot be synthesized in the body. Okay, and what are those? See, aromatic we have seen that phenylalanine and the tryptophan is your essential amino acid. If you look at the branched chain amino acid, right? Isoleucine, leucine, right? Lysine, valine. All these are also essential amino acid because branched chain kind of a structure, just think that way, is difficult to be synthesized in the body. That is why they are essential amino acid. What else? You know methionine, you know threonine, right? So you can make this sound similar, right? Methionine, threonine, These are also essential amino acid. Who else is essential? Your histidine. Histidine is also kind of an ring containing structure. It is also important, right? And now there is this arginine. Arginine. Arginine is essential on specific cases when there is a positive nitrogen balance. Only then arginine is essential. Only at that point of a time that makes arginine an essential amino acid. What is positive nitrogen balance? It is amount of nitrogen we take in the diet and the amount we excreted from the body, right? So if the intake of nitrogen and the excretion of nitrogen is balanced, is very well balanced in the body, right? If intake exceeds, right, then the excretion, it means body is having more of the nitrogen now, then that gives rise to the positive nitrogen balance. Positive nitrogen balance. Right? Now, other way round. If intake is less but the excretion is more, in case of the fasting, prolonged fasting case, right? When we enter that phase, then the muscles protein are being broken down to generate the energy for the survival. At that point of a time, intake is not there. But since the carbon amino acid have an amino part and the carbon part is the one which is gonna generate the energy. So nitrogen is being excreted. At that point in time, intake is less, but the excretion is more. That will give rise to the negative nitrogen balance, right? And arginine is essential in positive nitrogen balance. This way you can remember so many things together. Okay, so now let's move forward and let's take a look into the tryptophan. Okay, tryptophan is both glucogenic as well as ketogenic. It is an essential amino acid, right? And the various metabolites, why I picked this tryptophan? Because it is involved in so many pathways. It gives you so much. It gives you the serotonin. Via tryptophan, we can synthesize melatonin. NAD, NADP positive. These are what? These are the activated or coenzyme form of your niacin, your vitamin B3. That is also synthesized by the tryptophan. Acetyl coenzyme A. It gives you formate, which is responsible for the one carbon thing in the body. The alanine is helpful in the fasting state. Okay, so tryptophan breakdowns and gives you the alanine in the fasting state. And since niacin cannot be synthesized, niacin is a vitamin. It is an essential vitamin, okay? We cannot synthesize the niacin, but it can be synthesized by the body by breaking down the tryptophan. For every 60 gram of tryptophan, 1 milligram equivalent of niacin is being synthesized. Okay, so that is another very important point to be noted here. Right, serotonin and the melatonin are other important things other than the niacin. Niacin means we are synthesizing, the system is synthesizing, and NAD, activated form of niacin we are talking about, okay? Now, what else is synthesized by the tryptophan? Serotonin. Serotonin is synthesized in the pineal gland, and we know it functions as a biological rhythm, and it maintains the biological rhythm, and it functions as a neurotransmitter. Okay, what is melatonin? Melatonin, first of all, it is synthesized in an Ergen-Teflon cells of the intestine for the mast cells, platelet, and the brain. Okay, and what is melatonin doing? Melatonin is a vasoconstrictor, mood elevator, regulates the temperature, and is responsible for the gastrointestinal tract mobility. Okay, so these are very important things to focus on because deficiencies are important for all of us. Okay, before jumping into the metabolism, I would like to give you a broad picture. TRIP, that is also the L form in the body, can be degraded metabolized via two pathways. Okay, this is pathway number 1 and this is pathway number 2. Via pathway number 1, it is kynurenine pathway. It gives you many things. You form the formyl, you form the acetyl coenzyme A, you form the alanine in this pathway, right? So this pathway bifurcates in the end, making piconolate or piconolic acid and quinolinic acid. Okay, I apologize for my spellings. If they are wrong, please correct it by yourself. Okay, so this is one kynurenine pathway. Why the name so? Because one of the intermediate is kynurenine. This is the one pathway, and this pathway happens where? In the liver. Second pathway is the pathway where serotonin and the melatonin are synthesized. Right, and this happens majorly in the small intestine. So these are the two pathways. We will be taking both of them one by one in detail, and we will be majorly focusing on what is important. So tryptophan, right, there are two enzymes here, EDO and TDO. DO means these are dioxygenases. This is also known as dioxygenase pathway. Right, so these are both the dioxygenases which are present and which are regulated by the corticosteroids, interleukins. They stimulate this pathway, and via this enzyme, tryptophan is converted into N-formyl N-formyl kynurenine. That is why it is called as kynurenine pathway also, right? Then the formyl group is released, which leads to the formation of the kynurenine. Right? And formyl kynurenine, and then we make the kynurenine, which upon hydroxylation makes 3-hydroxy kynurenine. Right, and this kynurenine can be converted to 3-hydroxy anthranilate. And in this, at this step, what is being released? This is being released, alanine is being released, and this is taken down for the gluconeogenesis. Right, in the fasting state. Now, which enzyme is responsible? This is very important step we should be focusing on for the examination point of view. One, this is the point where we are forming the alanine. Enzyme involved is kynurenase enzyme, which requires vitamin B6. The activated form of vitamin B6 we all know is pyridoxal phosphate. This is the coenzyme activated form of vitamin B6. So if there is any deficiency of vitamin B6 will lead to what? Lead to inactivation of this step. This won't happen. So this will keep on accumulating. 3-hydroxyl kynurenine will be accumulated in the body, and that will be converted, then rerouted them to make the xanth uric acid, which can be eliminated from the body via the urine. Or we can say vitamin B6 deficiency led to the release of what? Led to the release of xanthurenic acid, right, in the urine, which imparts a yellow color to the dark yellow color to the urine. Okay, so this is one of the deficiency state. Let's say vitamin B6 is there, Let's continue further. 3-hydroxykynurenine is converted to what now? Is converted to 3-hydroxyanthralate, okay, and which is then converted to 2-amino-3-carboxymuconate semialdehyde. Now this is the point when it is going to branch off into the components we are looking for. We have already gotten the formyl, we have gotten the alanine. Now what is there? We have to get NAD positive, acetyl coenzyme A and picolinate. Major two pathways are this, quinolinic acid and picolinic acid. Okay, from the quinolinic acid, we get the activated form of niacin, that is NADP positive, right? And from the picolinate, we get other various things which we should not be worried about right now. And there is another pathway which makes alpha ketoadipate. From that we get the acetyl coenzyme A. This is the one pathway known as the kynurenine pathway, right? Because kynurenine is named because there is the formation of kynurenine. This is an intermediate. Enzyme, important enzyme is kynurenase enzyme, which requires vitamin B6. Deficiency will lead to what? Appearance of xanthurenic acid in the urine. Now, which is one is another pathway? This is the another pathway, right? In this, L-tryptophan is converted into 5-hydroxytryptophan. This is 5-hydroxy. TP is tryptophan. Right, hydroxylation is done and then you have 5-hydroxytryptophan. That 5-hydroxytryptophan can be converted into what? HT. What is this? 5H for hydroxy, 3 is tryptamine. So this is an amine now. So 5-hydroxytryptamine, right? And which can be converted it into, which can follow now two pathways. This and this. What is this 5-hydroxytryptamine? That is another important question. This 5-HT, 5-hydroxytryptamine, is another name for the serotonin. This is the another name for the serotonin. So in the question, in the exam, they can give you any of the name. You should be knowing it. 5-HT, 5-hydroxytryptamine or serotonin is one and the same thing, right? If the enzyme MAO is what? MAO stands for monoamino oxidase, acts upon it. It can be converted to 5-hydroxyindole acetic acid. 5-hydroxy, I for indole, acetic acid, okay, which comes out in the urine. When it will be converted into this? When their serotonin cannot follow the another pathway. What another pathway? When it cannot follow this second route, which I am labeling it by 2. Then excess of the serotonin will be converted into the hydroxyindole acetic acid. So you can put it in this way: if you have too much of the serotonin in the body for any of the reasons, there will be an increased or elevated levels of 5-HIIA in the urine, or the presence of 5-hydroxyindole acetic acid in the urine is indicative of the excess of the serotonin, right? Now look at the another pathway. What is going on? There is this enzyme acetyl transferase. Name is indicating it is doing the transfer of the acetyl group, or we can say we are acetylating the serotonin. So it becomes N-acetyl serotonin, because acetyl transferase added the acetyl group from it. And from where the acetyl group is coming? Acetyl group is coming from the acetyl coenzyme A. So this is acetylation. The next step is methylation. So enzyme is methyl transferase, which will add the methyl group to it, forming what? Acetyl methoxy serotonin. N-acetyl 5-methoxy serotonin. And what is so important about it? This is nothing. This is your melatonin. This is your melatonin. So we can say melatonin is what? Melatonin is synthesized from serotonin after the acetylation and the methylation. Acetylation, we need the acetyl group that will come from acetyl coenzyme A. For methylation, we need a methyl group, and there are two methyl donors in body. One is the tetrahydrofolate and another one is whom? The SAM, S-adenosyl methionine. SAM is the donor. SAM is the one who is going to donate, who is going to volunteer for the donation of the methyl group here, and we will be forming the melatonin, right? And what this melatonin will do? Melatonin inhibits the melanocyte stimulating hormone, it inhibits the adenocorticotropic hormone, and it also inhibits the ovarian function. See, this melatonin is different than the melanin. Mind this, melatonin, many a times in exam we get confused. Melatonin we are getting from the tryptophan, and there is this tyrosine, which I have discussed several other times, that is the melanin, and that is giving rise to the color to the skin, so leading to, so if there is any deficiency, it will lead to the what? Whitening of the skin and the hair, that is your albinism. So don't get confused, melatonin is your tryptophan, melanin is your tyrosine. So so remember this, trip is mel, tyre is mel. So what is the difference? Trip is onin and tyre is anin. So O and A, you have to remember this. Don't make this stupid kind of a mistake in the exam, okay? Now what else is about the regulation here? Not iso, again, I purposefully said that, IPRO, this is not iso, but this is isproniazid, right, which is a psychic stimulant. Why this is a psychic stimulant? Because it inhibits the monoamino oxidase. So it means what? There is the serotonin is rerouted and there is, you can say, increased amount of the serotonin, hence it is acting as a psychic stimulant, okay? And there is another drug known as reserpine. Reserpine is an antidepressant, right? It inhibits the synthesis of the serotonin, right? Now let's focus on the clinical correlate, why we have studied, where we are going to see all these things happening. So there are three diseases: Hartnum disorder we talk about, Carcinoid syndrome, and another name is the blue diaper syndrome or Drummond syndrome by the name of the scientist. So these are the three diseases, syndrome, symptoms we can see in relation to the tryptophan we talk about. Let's first focus on the Hartnum disorder. It is what? It is the defective absorption of the tryptophan. Tryptophan is an aromatic amino acid, but it is also a neutral amino acid, right? So if the other neutral amino acid plus tryptophan, which is also a neutral amino acid, is absent, right, from the intestine and the renal tubules because of the defective absorption. That is important, mind this. This is a defective absorption. You have it in the diet, but it cannot be absorbed by the body. Then it leads to the Hartnum disorder. Why this is happening? There is this gene SLC slice what? A19, 6A19. This gene synthesizes the BOAT1 protein. And this protein is important, right? Why? For the intestinal absorption of the tryptophan. So let's say if this is an intestine and you are getting the tryp from the food, it is there, right? Because this gene is defective, this protein is not there, so this absorption will not happen. this absorption will not happen. Rather, what will happen is tryp will be degraded by the intestinal bacteria into the tryp metabolites. We call them as indole metabolites. And later, towards the end of this session, I'll show you what those metabolites are. And they will be coming out. They will go to the systemic circulation directly, and then the renal circulation, and they will be excreted in the urine. And this excretion in the urine makes the diaper blue in color, known as the blue diaper syndrome. Okay. So, to revise it again, Hartnup disease is the genetic disorder because of the defective slice 6A. SLC is like, you can remember it, slice, okay. SL6A 19 number gene, right, which is responsible to synthesize the transporter protein, that is BOT1. Though it means the tryp is not absorbed, it is degraded by the intestinal bacteria, leading to a water-soluble metabolites which comes out in the urine, and they are indole compounds, giving the blue color diaper syndrome, because they are blue in color, and when the baby urinates, the diaper becomes blue in color. Okay. What will be the symptoms? Symptoms will be cutaneous photosensitivity because of decreased niacin, okay, and then neurological manifestation because of the decreased serotonin. Both are being, and there will be wide-based gait, intermittent ataxia, right? And diagnosis in the lab is done by the Obermeyer test for the indican. Indican is your tryp metabolite by the intestinal bacteria, and it comes out to be positive. For the treatment, NAD positive and lipid soluble ester of tryptophan is given. So this is your Hartnup disorder. The another is the carcinoid syndrome. Carcinoid syndrome is the tumor of what? Is of the Argentefin cells, which secretes the serotonin. So all of the tryptophan which is there will be taken up towards the second route, that is serotonin. So there will be no tryptophan available for the kynurenine pathway, right? This is what happens in the carcinoid syndrome, and that will lead to the symptoms like what? Symptoms like pellagra-like symptoms. Pellagra, we know, is a niacin deficiency disease, which is like 4 Ds: diarrhea, dementia, what else? Diarrhea, dementia, what else? Dermatitis, and finally, if not treated, taken care properly, lead to the death. So these symptoms will be seen because all the tryp is taken down towards the formation of the serotonin. What else is seen? Intermittent diarrhea and flushing, sweating and fluctuating hypertension. Diagnosis is made by chromogranin A, neuron specific enolase is used, or by the synaptophysin. Okay, so this is the carcinoid syndrome. Last one is Drummond syndrome, or you can say blue diaper syndrome, which is actually in conjunction with the Hartnup disorder, because in the Hartnup disorder we see the Drummond syndrome. Okay, before taking that, let's take a dive into the pellagra, since we are talking about this. Pellagra is all these symptoms we see. What do we see? The Cazel's necklace we see. So you can have any of these kind of these pictures in the question, or there can be these kind of disorders, skin disorders in any other parts of the body as well, right? Pellagra is 2 kind: diet deficient niacin, right, and poor ability to use the niacin from the diet. Both 2 cases we should be talking about. And especially the people, another question, especially the people who are on the maize, sorghum, sometimes they write the corn. Maize is corn, sorghum is jowar. Okay, corn. We should be knowing the keywords: corn, jowar, right? Maize is corn, jowar is sorghum. What they have is they eat too much. If they eat too much, what it has is there is the leucine in these diet that interferes with the tryptophan metabolism. So it means it is the person will be showing the pellagra if he is on, like, for prolonged time he is eating all these things, which is leading to the 4Ds of the pellagra: diarrhea, dementia, dermatitis, and death. Now another thing here, look from the middle. In relation to the tryptophan, there are three things we have studied. One is the vitamin deficiency, that is vitamin B3 or the niacin deficiency, Hartnupp's disorder, carcinoid syndrome. All these things what we will be finding, the patient having the pellagra-like symptoms, but you have to look why that is happening. So you have to look for the clues to pick the correct answer, which actually in haste we pick the wrong answer. So carefully read it. Hartnup syndrome is failure to absorb the tryptophan. Carcinoid syndrome is because tryptophan is there, but all the trip is taken towards the formation of the serotonin and none is left for the niacin. And this is the failure to synthesize the niacin. If body cannot synthesize the niacin from the tryptophan, then it will lead to the pellagra-like symptoms. So you have to be very careful what the examiner is asking you. Okay. Now, I told you about that if the BOT1 protein, transporter protein, is not there in intestinal bacteria is going to degrade them into the water-soluble compounds. So these are those compounds. What we form is we form the indigo blue or indican or the indoles. So tryptophan is converted to these metabolites, right, and they impart blue color, hence the blue diaper syndrome. Right, it is a metabolic disorder, inborn error, and autosomal recessive in nature. So this is your blue diaper syndrome, right? And it is usually, when we are saying diaper syndrome, it is obvious we are talking about an infant, okay? So you can have something like this in your clinical presentation or maybe in your exam. This is the Obermeyer test. Obermeyer test is positive for the people who have this blue indigos or indicans in the urine. But see, there are three colors. That tells the how much it is being excreted in the body. It is that is just a range from the light blue to the jet black. It can range from anywhere from 2, 3, 4. So this is the Obermeyer test. Why I'm using the pictures? Why I use so many things? Because we tend to forget the words, but we definitely remember the pictures. So this is my practice to use lots of picture. Let's quickly take few of the questions. The first question is, the patient is suffering from carcinoid tumor and has elevated of 5-hydroxyindole acetic acid in the urine. 5-hydroxyindole acetic acid is metabolite of what? Is it a metabolite of histidine? No. Is it a metabolite of niacin? No. Is it a metabolite of melanin? No. It is a metabolite of serotonin. In haste, don't pick niacin or melanin. Why? Because they sound similar to the one we have studied in the tryptophan. So it is HIAA is serotonin. Middle-aged lady has developed rashes on the exposed part of the body. Developed rashes, so dermatitis. And history of diarrhea. Second D. Upon investigation, family member witnessed her disordered memory. Third D. Dementia. She is on isoniazid therapy. Very good. Isoniazid therapy is for tuberculosis and that prevents the tryptophan from the diet, right? What would you predict about her? The niacin deficiency, because isoniazid therapy inhibits this. 5-fluorouracil also inhibits the absorption of the tryptophan from the diet, okay? Which defective gene is responsible for defective neutral amino acid transport in intestine and the kidneys? Responsible for the Hartnup disease with pellagra-like symptoms. So which amino acid we are talking about? Hartnup disease is in relation to the tryptophan, which is the neutral amino acid, right? And which gene is defective? This is your splice gene. So this is your splice gene, all are splice genes. 6A18, 6A19, 36A, no, it is 6A. Which 6A, 18 or 19? It is 19. Remember this combination. I guess you remember it very well. Okay, so this is 6, this is 9. Don't forget this part, okay? Patient is detected with zinc urinic acid in the urine and tryptophan loading test is done, which it is due to the deficiency of what? When we did it? So here this is the part. Let me erase everything else so that you can see it very clearly. Right? In this case of deficiency of vitamin B6, what we do is we do the tryptophan loading test. Trip loading test. And this is obvious because of vitamin B6 deficiency or enzyme deficiency. This is not going to follow towards the formation of 3-hydroxyanthranilate, right? Whether it will make the zinc uric acid here, and that is excreted in the urine. So this was your tryptophan loading test. Right, now read the question again. The patient is detected with zinc urinic acid and tryptophan loading is done. It is due to the deficiency of what? Thiamine? No, it is related to the beriberi disease. Biotin? No. Folate? No, it is vitamin B6 pyridoxal 6-phosphate, right? This is the answer to it. Thank you very much and good luck. And please keep me posted. What else would you like me to release the video on YouTube? Good luck.